Any part of it.
Action.
Photosynthesis. Alright, we'll start with the crank. Turning the crank represents photons of light boosting the energy level of an electron. The red and yellow gears represent a pigment molecule that transfer energy to a P6A molecule in the reaction center complex. The red swing arm represent the path that electrons, provided by splitting of water, shown by sticky note arrows, take as they transfer from P6A to the primary acceptor, which is the green boot. Electron transport chain, which is this blue thingamajig. Electrons, represented by the metal ball, travel down the electron transport chain and to the photosystem 1. In the first stage of the electron transport chain, electron carrier plastoquinone, PQ as it's known, or the blue slope, transfers electrons to the cytochrome complex, which is the red slide. The cytochrome complex produces ATP, and these are the cheese shapes you see here. And as electrons fall to a lower energy level. And the proton plastocyanine, or PC again, which is at the end of the red slide, this transfers electrons to photosystem 1, which is this thingamajig right here. Alright, so now we're at photosystem 1. Electrons are transferred to P700 right here, and the ball strikes the blue tower, and they're boosted to a higher level by photons. And the excited electron is then transferred to the primary acceptor, which is the red platform atop the blue tower. And now we go on to the electron transport chain. Electrons fall down the electron transport chain, down the blue tower, carried by electron acceptor ferredoxin, which is FD, represented by a blue lever right here. And the electrons are transferred by NADP plus reductase, which is the yellow cup, which facilitates the reduction of NADP plus and NADP pH, represented by the descent of the red cage down the thingamajig. The NADPH is released in the stroma, and then it goes to cyclic electron flow. In cyclic electron flow, electrons are diverted from the primary acceptor, the red platform, to the cytochrome complex, the red slide, to produce ATP, rather than travel down the electron transport chain to produce NADPH.
The Calvin cycle is in the stroma, represented by the separate sheet. Phase 1. Fixation. 3 carbon atoms enter the cycle and bond with the 3 5-carbon sugars, RUBP, to form 3 6-carbon sugars that separate into 6 3-carbon sugars, 3-phosphoglycerate. Phase 2. Reduction. 6 ATP molecules from the light reactions are then used to phosphorylate the 6 phosphoglycerates from 6, 1, 3 biphosphoglycerates that are then reduced by 6 NADPH to form 6 glyceraldehyde 3-phosphate G3P, one of which is then released by the Calvin cycle. Phase 3, RUBP regeneration. The remaining 5 G3P then proceed through a series of redox reactions using 3 ATP to rearrange the 5 3-carbon sugars into 3 5-carbon ribulose biphosphates that restart the cycle.